and welcome to the protein synthesis screencast. We are moving from DNA to RNA to proteins, and we're going to start by talking a little bit about DNA. The big picture I want you to take from this timeline here is that the discovery of DNA's structure, this double helix structure that we all know and definitely love, it took the work of many different scientists over many decades to arrive to the level of understanding that we are at today. I pointed out a few scientists to you in class, Watson and Crick, as well as Chargaff, and Rosalind Franklin and Maurice Wilkins. Maurice Wilkins, Watson, and Crick were actually the recipients of the Nobel Prize. They were awarded after discovering this double helix shape of DNA. This video, along with all of the other videos that are embedded within this PowerPoint and included in other resources from class, like the stop motion animation slide deck that I gave you, as well as the outline, I gave you some resources on there. You can find links to this and all of them on the post on Google Classroom where I posted your review guides. I think the title of the document is something like Digital Resources, but jump over there if you'd like to watch some videos or play with some interactives, or I think there's even a game on there too to help you with your level of understanding of protein synthesis. We're not going to watch that. Okay, so in your notes in class, we did this as practice to label these different structures within DNA, but I'll just kind of review it for you. So we're looking here, this is our double helix shape, almost like a twisted spiral staircase. If we untwisted this double helix shape, it would look like this, almost like a ladder. The outsides of the ladder are the sugar phosphate backbone, and this is almost just like a little zoomed in portion of this, almost as if we took a chunk right here. The sugar phosphate backbone is called that because it contains the 5-carbon sugar, deoxyribose, and the phosphate group of the individual nucleotides, those are the monomers of DNA that compose this whole molecule, this polymer. I'm also going to point out to you that the rungs of the ladder are made of base pairs, and they're attached by hydrogen bonds, weak hydrogen bonds, compared to the covalent bonds that hold together the sugars and phosphates of that backbone. I want to point out to you up here that DNA stands for deoxyribonucleic acid. As a reminder, DNA is a type of a nucleic acid. Remember, a nucleic acid was one of the 4 major macromolecules that we talked about in class. And then deoxyribo, we're referencing down here this 5-carbon sugar that's a part of those nucleotide monomers that hold together or that compose that nucleic acid. So let's talk about those base pairs. These are the pairs that are held together again by hydrogen bonds, creating the rungs of those ladders. In DNA, we have thymine, adenine, cytosine, and guanine. Though most often, you're just going to be writing them represented by their first letters. There are also 2 sets of base pairs. Adenine bonds with thymine and cytosine bonds with guanine. Between adenine and thymine, you will find 2 hydrogen bonds, and between cytosine and guanine, you will find 1, 2, 3 hydrogen bonds. I like to remember those base pairs with this little mnemonic device, always together, great couple, though I'm sure that there are other ways that you could try and remember them. I also want to point out to you that DNA strands are anti-parallel. That means that these 2 strands are actually running in opposite direction of one another, and they're each assigned a 5' and a 3' end. That 5' and 3' end is in reference to the location of the carbons within this deoxyribose sugar. So we're not going to get super specific on that. I just more care about you taking that message away that they're running in opposite directions. So you can almost look like, let's look at this nucleotide right here. You have this phosphate group, the 5-carbon sugar in the center, and then this nitrogenous base, guanine. Same thing if we look up here on this particular side. Phosphate group, 5-carbon sugar, the nitrogenous base. You can see the oxygen at that almost like this is a pentagon looking like a house. It's at like the peak of the roof. That's where the oxygen is. If I look over here I want you to notice that now those peaks, that oxygen and the peak of the roof of the house of the deoxyribose is now facing in the opposite direction. And that 3', 5' is in the opposite order compared to over here. It's like a highway. The cars, they're parallel, those highway lanes, or those street lanes, they're parallel, but the cars are always running in opposite directions. In class, we labeled these, so I had you discuss at your tables. You don't have this in your notes. I'm going to pop up the answers. You can press pause or just watch them show up. First of all, we have the pink phosphate group, usually represented by a circle. You have that 5-carbon deoxyribose sugar. We have the sugar phosphate backbone, again composed of the 5-carbon sugar and the phosphate group. Then we have a full nucleotide, that's the monomer of DNA. DNA is a polymer. And then I use my knowledge of my complementary base pairs, A and T, C and G, to fill in the correct complementary base. And again, we have those 2 lines to represent 2 hydrogen bonds and 3 lines to represent 3 hydrogen bonds between those base pairs. Next, you needed to interpret the differences and some similarities between DNA structure and RNA structure. DNA and RNA are both essential molecules in protein synthesis. And knowing the differences between the two is also going to be helpful for you knowing their roles, their numerous roles between those two processes. So I'm going to point out some important ones to you. First of all, I want you to look at the name. We already addressed deoxyribonucleic acid is what DNA stands for, but now we have ribonucleic acid is what RNA stands for. RNA is also a nucleic acid, just like DNA. And I told you that the deoxyribo portion up here is referencing this 5-carbon sugar that we find in DNA called deoxyribose. In RNA, we have ribonucleic acid, ribo referencing this 5-carbon sugar, a little bit different from the one found in DNA, and it's called ribose sugar. Okay. I also want to point out to you DNA is double-stranded. You can see those 2 strands here, while RNA is single-stranded. That means while DNA has these base pairs within its molecule, RNA does not have base pairs here, though we do have some base pairing rules. They just have these little single nucleotides creating that one strand. The nitrogenous bases found in RNA are cytosine, guanine, adenine, and uracil. So that means that we're going to adjust our base pairing rules in RNA compared to DNA. DNA has thymine, cytosine, guanine, and adenine. And we know that adenine and thymine bond together, cytosine and guanine bond together. Cytosine and guanine bond together in RNA just like they do in DNA. But now, since there's no thymine present, adenine bonds with uracil. So the base pairs in RNA are A and U, C and G. All right, let's move on to this. This is a little C for you. Didn't mean to pop this up a little bit early, but I wanted you to practice writing the complementary strand to each DNA molecule that I presented to you. So here's the first strand. Again, starts with 3', ends with 5'. So my complementary strand is going to start with 5' and end with 3'. I'm going to pop up the other answer here so you can check your work, and we'll just keep moving forward. Big picture, why is this applying to you? Why are we covering this in class? Well, first of all, let's identify a few vocab terms. A gene, so maybe you've heard this before. A gene is just a segment of DNA. DNA is a long molecule, a big molecule, a macromolecule, and it contains many genes within it. Now, a single gene is just a section of DNA, and that gene codes for a specific protein. Each unique gene has a unique set of bases. The way that the A's and C's and T's and G's are ordered, it's unique to each gene. And that unique set of bases are going to code for the production of a specific protein. That's that protein synthesis that we're going to be talking about. And those protein combinations determine our unique characteristics or our phenotype, our physical characteristics. That is what we look like is an expression of our genes found in our DNA. So again, we have a cell. Our genome is just the sum of all of our DNA, all of our genetic information. It's made up of chromosomes, and our chromosomes are made up of tangled DNA and all these other structures that we'll get to a little bit later in the year. But if I look at a segment of DNA, there are many genes within DNA. And again, you can see each one of these little segments, each one of these little genes have a different unique set of bases, which means that they code for different proteins. And those proteins allow for the expression of our genes. All right, here we go, nitty gritty. Protein synthesis. We're going to do a little bit of an overview before we get into the specifics. There are two major parts of protein synthesis. There is transcription. That's the process of taking the message of DNA and transcribing it to mRNA. And then translation is the process of taking that message of DNA from RNA and translating it from nucleic acid language to protein language. This flow of genetic information is known as the central dogma of biology. DNA to RNA to proteins. Dogma means belief. So transcription occurs where the DNA is located in our cells. And I'm saying our cells because we're talking about eukaryotic cells. We contain eukaryotic cells because our cells contain a nucleus inside of them. Any cells that do not have the ability to contain a nucleus in them are prokaryotes. And they undergo protein synthesis a little bit differently than we do. So again, we're just focusing on eukaryotes for now. Though when we talk about cells next unit, you'll understand this difference a lot better. So transcription occurs in the nucleus. DNA has to stay in the nucleus. It's safe and protected in there. It does not leave the nucleus. So that message of those genes needs to travel out of the nucleus and into this space out here called the cytoplasm, where translation happens, where we're going from that message of RNA to a protein. And just as a reminder from our last little slide, genes, those segments of DNA, code for proteins. And proteins determine our phenotype, our physical expression of our genes. So if I'm looking down here as this example, so this is a little chromosome. I have a little segment of DNA in here, and it's the gene that determines flower color or contributes to flower color. I have this unique little DNA sequence that's going to code for a particular protein. That protein helps to make pigments that determine the color of the flower. Let's talk steps of transcription. Again, we're going from DNA to RNA, and this is happening in the nucleus. There are three main parts of transcription. Initiation, where we have this special enzyme called RNA polymerase. What it does is bind to the promoter. The promoter is just a segment of DNA, and it indicates the beginning of the particular gene that we're transcribing. So RNA first binds to that promoter, and then it begins to unwind DNA. I want you to think of RNA polymerase as almost like the zipper part of your jacket, okay? So we're unzipping these rungs of our DNA ladder, breaking again those weak hydrogen bonds compared to those stronger covalent bonds in our backbones. And it's exposing these single strands, and those are used as templates to do the next part, which is to begin to build this new RNA molecule. This RNA molecule contains the complementary bases to this template strand. But as a reminder, we're building an RNA molecule. So instead of using thymine, we're using uracil in this new mRNA strand. There is no thymine in RNA. I also want to point out to you here that this RNA strand looks almost identical to the coding strand, which would be this strand, except for, again, the T's are replaced with U's. RNA polymerase continues to move down DNA, unwinding and pairing complementary nucleotides until it reaches the terminator. So just like the promoter indicates the start of the gene, that start of transcription, the terminator signifies the end. Once RNA polymerase arrives to the terminator, the mRNA is now complete. It detaches, as does RNA polymerase, and now RNA goes off to be edited in the nucleus.